Thank you for attending this webinar entitled The Fibrillar and Oligomeric Constructs of Neurodegenerative Disease-Associated Peptides and Their Utility in Disease Modeling. My name is Ariel Louwrier, and I'm the president of StressMarq Biosciences Incorporated based in Victoria, Canada. Over the last few years, this company has begun the process of producing and characterizing a range of fibrillar constructs of prion-like proteins. We're going to elaborate on this after an introduction and examine some of the fibrils, filaments, and oligomers that we have made, as well as some of their characteristics. We will then go on to look at aspects of sonication. Sonication is one of the primary methods that is used to prepare fibrils for use in vitro or in vivo experiments. And very little is known about what actually occurs during this process. Finally, we'll look at some aspects of fibril conjugation before concluding this event. Now, by way of scientific background, alpha-synuclein is a relatively small protein, approximately 15 kilodaltons in size, whose physiological function is not well understood. It can act in a prion-like manner and can generate a large family of different sized oligomers, again, with different properties. They also vary in their structural aspects. Now, oligomers can then go on to generate fibrils further down the pathway, also known as aggregates. Now, these fibrils are toxic to cells and are now known to be processed by cells themselves by virtue of C-terminal truncation. Alpha-synuclein can generate Lewy bodies and neurites present in Lewy body dementia, and we'll have a look at that shortly. The aggregates are present in Parkinson's disease and kill brain cells. They can be transmitted from cell to cell, causing the disease to spread throughout the brain, which results in, of course, the disease progressing. And on top of it, the aggregates themselves can cause other proteins to continue or to start to aggregate, including related proteins such as gamma-synuclein. Synucleinopathy is a very broad term associated with neurodegenerative diseases where alpha-synuclein plays a key part, have no known cure, but incorporate diseases such as Lewy body dementia, Parkinson's disease, and multiple systems atrophy. Now, in this schematic taken from Roberts and Brown cited below, you can see how some of these oligomers may result from the initial monomeric species. On the far left-hand side, you see the blue monomers, which then aggregate together to form a variety of different sized and type oligomers. Some of these may be what is referred to as off-pathway, which means they're not on the pathway that ultimately forms the amyloid fibril. They could be toxic or non-toxic. However, those that are on pathway continue to aggregate, and the exact species and their structures, as well as their characteristics, are not well understood. However, they go on to form protofibrillar oligomers, which are still soluble in nature, and these are then assembled into the amyloid fibril at the far right-hand side, and generally speaking, these tend to fall out of solution. That drives the process towards the right, despite the other components of this being in equilibrium with each other. Now, in this slide, we're able to identify some of these key structures that alpha-synuclein can result in. The round, dark spherules are labeled as Lewy bodies, and although initially thought to be just filled with aggregated alpha-synuclein, we now know that it contains other aggregated proteins and even membranes and organelles. The neurites are a completely different structure, also formed with alpha-synuclein, and are string-like in structure and are labeled on the right-hand side. In terms of how alpha-synuclein aggregation actually affects the brain in Parkinson's disease, this slide shows the substantia nigra, which is a part of the brain that is associated with movement. And there, the monomers as you can see in the middle of this slide, moving to the right, aggregate into oligomers and then protofibrils, and then before final assembly into fibrils. And these interact with dopaminergic neurons, killing them. There's a high level of toxicity associated with them. The cells die, and as a result, we see the movement disorders that we associate with Parkinson's disease. In terms of the protein itself, it has a basic hairpin type structure with a fair chunk of alpha helix on the N-terminal side and about halfway down into the protein. Most importantly here, labeled in blue, is the fibrillization core, which is a sequence that is prone to change, and that change results in the alpha helix changing into a beta sheet structure. On the far right-hand side, there is a labeled moiety, serine 129, which gets phosphorylated when aggregation occurs endogenously in living systems. And that is one of the key gold standards that is used to monitor the evolution of pathology. One other area that is of note, although there's a lot of labeled moieties on this slide, is alanine 53. That is the source of the familial Parkinson's disease. The A53T mutant is in fact one of the products that StressMarq has made and is quite an aggressive form of the alpha-synuclein protein in terms of aggregation. Now this slide gives you an idea of what happens during the aggregation process. You can see that right at the beginning, start with the alpha helical structure, and it goes through a variety of changes depending on its energy states, and results in these beta pleated sheet structures. Now that's particularly important because we can use those beta sheet structures and bind something to them, in this case Thioflavin T, which generates a fluorescent signal, making it a very simple assay and an easy way to measure aggregation in vitro. And this slide shows you that process. The blue line shows the increase in fluorescence as Thioflavin T binds an increasing level, an increasing concentration of alpha-synuclein fibril in a seeded reaction, where fibrils are added to monomers, essentially catalyzing the reaction of monomer to fibril. And you can see in the controls at the bottom in red the active aggregate on its own, as well as monomer on its own. Now, if you were to, for instance, wish to interfere with the process of aggregation, then this is the type of assay that you might employ, and you would be looking to flatten this particular curve or stop the aggregation reaction altogether. In terms of tau, this is a larger protein. It's about 46 kilodaltons, but has a whole variety of isoforms that we'll look at in just a moment. Tau is known to stabilize and promote the assembly of microtubules in neurons, and can also act in a protein-like, prion-like manner. Phosphorylation of the protein results in the transformation of what we might think of as normal physiological tau into paired helical filament tau, and ultimately into neurofibrillary tangles, which are referred to as NFTs. Now, NFTs may be toxic to cells. It is not entirely clear if they are actually a repository for damaged tau, or if they are toxic in their own right. There is evidence of both. However, here we can see some of the different types of tau. Circled at the top is the 2N4R, which is essentially the full-length tau, which StressMarq uses to make fibrils with, but we also use some of the more attenuated versions, which are composed of the microtubule binding domains, which you can see on the right-hand side are 1 to 4. Now, as hyperphosphorylated tau leaves the microtubule, the neuron transport systems begin to disintegrate, and we'll look at a schematic of that in just a moment. But essentially, the microtubules are pathways by which essential nutrients are transported downstream, and if those microtubules are disrupted, then the nutrients cannot be transported to their final destination, and the cells begin to die, and as a result, of course, the brain's functions are impeded. Now, tau is particularly interesting in terms of Alzheimer's disease, and as brain function is impeded, of course, the symptoms that we associate with Alzheimer's increase quite dramatically. And in this schematic, you can see in the top insert the microtubules with physiological tau associated with them form these nice rigid pathway structures. However, once the hyperphosphorylation occurs, the tau leaves the microtubules and the microtubules are severely disrupted. You then get in the bottom right-hand corner the paired helical filament construction, and then the assembly into these clusters referred to as the neurofibrillary tangles, and the cells begin to die. Now, in terms of the actual products and tools that are provided by StressMarq, the reason we want to go through this in some detail is to highlight some of the differences between the different versions of the same protein. There is no one-size-fits-all. Now, in terms of monomers alone, we have what we call type 1 and type 2. We'll be looking at that in a little bit more detail in terms of the fibrils, but essentially type 1 will form type 1 fibrils and type 2 will form type 2 fibrils. Oligomers can be present in a variety of different ways depending on how they're made, but generally are assumed to be roughly spherical, about 20 or 25 nanometers in diameter, and we'll look at that a little bit further on in this webinar, or as small twisted pigtails. They look like small, highly twisted, about 25 nanometers across fibrillar structures. We then have another class, which we refer to as filaments, and these are still soluble in nature and do not have the properties associated with the fibrils. However, under EM, essentially look the same. And that can cause a great deal of confusion because these, as you will see later on, are not capable of seeding, although fibrils are. And then finally at the bottom, the fourth class here are the fibrils themselves. We have type 1, 2, and 3, where 3 is in many ways an amalgamation of 1 and 2, and we'll be looking at that shortly, as well as different types from a species perspective, mouse and human, and then also mutant types, such as the A53 mutant. And these all have differential properties. So here we can start with human versus mouse alpha-synuclein preformed fibrils. And you can see that they look very much the same. The sizes are indicated here on both slides, and they're about ranging from about 30 to 70 nanometers in length, but overall look approximately the same. However, mouse alpha-synuclein preformed fibrils tend to be a little bit more aggressive in the ability to create phosphoserine 129 pathology in vivo and in vitro. Now, as an overview, here's some electron micrographs of type 1, 2, and 3 fibrils, as well as filaments. And type 1 and type 2 have been studied. Type 3 has not been well studied. But what we want to really illustrate are some of the differences as well as the similarities between these constructs. So clearly, to the naked eye, these all look rather similar. Bear in mind that the size bars in the bottom right-hand corners of each individual micrograph is not always the same size. But you can see in some cases, particularly with type 1, that there are some duplexes involved here, where clearly strands are associated with each other. That is also supported by the literature. You can also see that in the type 2, but essentially all of these different types look very similar. When we move to oligomers, things begin to change quite dramatically. So here we begin to see these roughly spherical globules that are associated with the structure known to occur when alpha-synuclein is induced with dopamine, probably the most physiological relevant chemical from the brain perspective. And these are very, very different. They're really quite stable. They're far more stable, for instance, than fibrils are to STS page. If you run an STS page of the dopamine-induced alpha-synuclein oligomers, you see the size ranges that you would expect them to be at, generally quite large. However, with fibrils that are structurally much, much larger, they're all reduced to a monomeric form when STS page is used. Comparing type 1 and type 2 alpha-synuclein fibrils is rather simple. Here you can see the aggregation reaction that we talked about earlier, and the purple line represents a type 1 fibril being generated through a seeded reaction. And you can see that in a situation here where all concentrations are the same, that the level of fluorescence, i.e. the evolution of beta sheet that's associated with a type 1 fibril, is very high. And it goes up rapidly, and then it begins to plateau off, whereas a type 2 fibril has a much lower fluorescence signal, i.e. the beta sheet content is significantly less. And in this case, and we have seen this consistently over the years, approximately 20 to 25% of the signal you would associate with a type 1 fibril. The differences continue to be manifest when you add these to primary cells, in this case rat primary cells, neocortical primary cells. And on the left-hand side, you can see type 1 fibrils are used, and they generate a lot of phosphoserine 129 pathology, shown here in green in the middle on the left-hand side. Whereas if you do the same with a type 2 fibril, you generate almost no pathology whatsoever. As a result, you can imagine that the type 1 fibrils are responsible for the overwhelming majority of the research and publications that have been done worldwide over the last few decades. Very little in that regard is known about the type 2 fibrils, and because they don't generate the pathology, they have definitely been less popular and essentially absent from the literature. However, the evolution of pathology is only one component. Toxicity aspects are another component, and anybody that is doing research into this area needs to be aware that there is more than one type of fibril, and that the effects of adding these fibrils to living systems can be diverse. Otherwise, they may well be looking at only a half or less of the entire picture. Here we see the effects of adding the A53T mutant, which I already mentioned is quite aggressive, to Shy5 cells. And you can see that using, in green, a total alpha-synuclein antibody that can also measure aggregate formation, that you get increased aggregates that result quite immediately in high levels of cell death, and a dramatic increase also in red in the phosphoserine 129 signal generation, which then also levels off as the cells begin to die. The same is true with type 3. So type 3 is a fibril that looks very much like a type 1 fibril, but has endotoxins levels that are significantly lower, which would be more those that you might associate with a type 2, but has the structure of a type 1. And here you can see the phosphoserine 129 signal in red also increases quite dramatically in the initial stages after being added to the cells, and then begins to fall off after that as cells begin to die. However, if we take the filaments instead of the fibrils, which remember under EM look essentially the same, so if you were using that as a quality control, you might well be easily led to make this mistake. If you add these to Shy5 cells, you'll note there is no seeding, there is no increase in phosphoserine 129 signal. And this is supported by the fact that in vitro, the filaments, which again, soluble precursors of the fibrils, do not show any capability of seeding. And then finally, the same is true for oligomers. So here we have dopamine-induced oligomers, again added to Shy5 cells, and the red signal being the evolution of phosphoserine 129 signal. And we do not see an increase in phosphoserine 129. Again, this is supported by the literature, where it has been shown that these are quite toxic, but do not have a seeding capability. We then have also worked with a variety of different partners, and one of them is a CRO based in Vienna called QPS Austria GmbH. And they did some testing for us using a couple of systems, and they looked at some of our type 2 preformed fibrils and the A53 preformed fibrils, which are a type 1, again quite aggressive, as well as some of our oligomers and controls. And you can see that the toxicity associated with the oligomers is very, very prevalent in these experiments. So in the MTT assay, you can see the toxicity is far more of the oligomers than of the fibrils, whether it be type 1 or type 2. That is then borne out by the LDH assay on the right-hand side, where you can see again in orange the oligomers stick out quite considerably as being that much more toxic. So we get on the left-hand side, we can see in the MTT assay, you can see this decrease of about 20%, but about 50+% when it comes to the oligomers. Now, it may be, of course, that the fibrils either generate oligomers on their own and that they are a toxic species, or that the toxicity is just associated with the fibrils themselves. That is not something that is well understood at this time. Bear in mind again, when we looked at that early schematic, that a lot of these fibrils are in equilibrium with other forms of alpha-synuclein. So what happens when you stick these into rodents? In this case, we have used type 1 PFFs that have gone and been injected into rats. And what's of particular interest here is that the evolution of the phosphoserine 129 pathology is generated after just 30 days. Compare that perhaps with transgenic animals, where evolution of pathology may not arise for many, many months or even over a year. And in this case, we also see that not only is the pathology generated on one side of the brain where you might inject this material, but also on the other side of the brain. So the pathology is transmitted throughout the brain as well. There are other forms of synucleins as well that we're not going to spend a lot of time on, but are relevant in terms of the product offering. This includes beta-synuclein and gamma-synuclein. And again, looking at them under electron microscopy, they don't really look very different from any other fibril. And The one interesting thing that we'd like to highlight in particular is beta-synuclein. Now, beta-synuclein doesn't seed on its own. You have to treat it quite harshly in order to get it to seed. And this graph here shows a comparison of different seeding events. The blue line on the left-hand side shows a typical alpha-synuclein seeded reaction. However, if you look at the red line on the bottom, this shows beta-synuclein fibrils with alpha-synuclein monomers, and the green line that it overshadows is a beta-synuclein fibril with a beta-synuclein monomer. So not only does beta-synuclein not have the capability of seeding a reaction of its own monomer, but it also cannot seed alpha-synuclein monomer either. Compare that with alpha-synuclein monomer on its own, unseeded. This is best illustrated in the insert on the right-hand side with the dotted blue line, where alpha-synuclein, after a while, begins to increase in fluorescence. And that is normal because after a certain point, alpha-synuclein monomer will self-nucleate and begin the elongation process into oligomers and fibrils. What this shows us is that if you want to control, then beta-synuclein fibrils may be the best choice because A, they are fibrillar in nature and have a very similar structure to alpha-synuclein, but they are incapable of seeding, whereas monomers at a certain point will self-seed and begin to generate signals. Moving over to tau, we can see on the left-hand side in this micrograph the full-length 2N4R P301S mutant fibrils, and these are quite highly branched. This is also a side effect of the heparin induction that is used to create these aggregates. On the right-hand side, less of a branch structure when you use the K18, which are just the repeats that we showed you earlier in the webinar. So what happens if you take the full length tau and you place it in Chi5 cells? So these would be the aggregates that are added, and the green signal here is using a total tau antibody, and you can see that the aggregates get quite prolific, but again, as with alpha-synuclein, the cells begin to die rather rapidly as a result. And so the evolution in red of the phospho signal associated with moieties 202 and 205, which is largely in the literature regarded as the AT8 site, doesn't really have a chance to evolve. When we entered into another partnership with Remind, based in Belgium, they injected the K18, so the smaller version of tau, into mouse brain, and they were able, using the same AT8 site, to detect phosphorylation at the injection site. So it does look like the K18 is able to seed in a in vivo situation. Now, in our quest to try to make tools that are as relevant as possible to what is happening in human disease, one of the key aspects from a structural perspective is to generate the paired helical filaments. Now, the K280 mutant has been shown in the literature to spontaneously generate these paired helical filaments. Now, sometimes this can be quite difficult to identify on electron microscopes, and you need to be an expert to do so. But in some cases, we can see in the micrograph on the left that there are a couple of structures there that are more likely to be duplexes. Approximately middle on the right here, you can see that there are two different fibrils, essentially a duplex associated with each other. You can see the same here as well. However, interestingly, on the right-hand side, we can see that the fibrils, the K280 fibrils, are capable of seeding themselves. Again, as we continue to drill down, one of the elements that becomes more and more important from a drug discovery perspective is how does one disrupt the evolution of fibrils? And to do that, we have another construct, which is a much shorter version, which represents the amino acids that are responsible for generating the core of the fibril. And there are two versions of this. One is a cysteine mutant that allows you to play with some redox parameters, but essentially they are the same thing. And both of them form these fibril constructs. And again, these would be of interest to anybody that is interested in trying to manipulate elements of the core of the tau fibril. As we continue that quest to try to generate those structures that we see in the brain, we have also tried to take the full length, and now instead of using heparin as an induction method, to grow it on a linear anionic scaffold. And this is what you see on the left-hand side. So these filaments here can much more clearly be identified as having a duplex twisted structure. The longer filament in the middle of the left micrograph shows that reasonably clearly. And then finally, on the right-hand side, we have generated fibrils from tau that were made in a baculovirus expression system. And these are particularly interesting because they don't require any induction whatsoever. And these will have, by virtue of their post-translational modifications that are inherent to the system, will begin to self-aggregate on their own. Here we can see that these tau preformed fibrils made in the baculovirus expression system are able to seed, although because the event is self-seeding, from an inherent perspective, you see the green line in the middle also rising as the red one does just at a lower level. In particular, when we're talking about post-translational modifications, we know that tau is phosphorylated reasonably heavily, and of course, during the process of Alzheimer's disease, becomes hyperphosphorylated. So here we can see that just generating the construct in baculovirus results in approximately 45 or so of these moieties undergoing post-translational modifications. The one that's highlighted in the bottom left-hand corner is associated with the one site that, again, is used most prolifically as identifying the evolution of tau pathology. With reference, this is called the AT8 site, and it's serine 202 and threonine 205. Now, we're not going to talk about antibodies a great deal. There's just one or two slides here that I think show them from a perspective as being quite interesting tools. And in terms of expressing tau in iPSC-derived neurons, you can see clones SMC 607 and 608 showing really very, very nice, clean imagery against tau. So these would be total tau clones, being able to identify both monomeric and oligomeric forms, and compared to the total tau antibody from DACO in red, and Thermo Fisher 88 clone itself in the bottom left-hand corner. Now, in terms of actually being able to identify those phosphosites at 202, 205 in tau, again, this is the gold standard for measuring the pathology generation of tau fibrils. You can see that clone SMC 601, or sorry, I should say catalog number SMC 601, clone AH36, on the left-hand side doesn't show you very much in the control, but on the right-hand side, in the mutant cells on the right-hand side, shows you the phospho-tau very, very well, whereas the 88 clone below does a poorer job of that, although seems to have the same specificity. Now, we've talked a lot about alpha-synuclein and tau, but those are not the only proteins that we work with. There are a range of others, and I'm just going to mention one or two here. One is transthyretin, TTR, again, a whole range of fibrils that are generated there that are capable of seeding, and we do generate two of these mutants. One is the L55, P filament, and the other mutants, the Y75F, and both are capable of seeding. Now, moving on to the sonication questions, here we highlight in this slide the best practices associated with the sonication of alpha-synuclein. And this is deemed as completely mandatory because if sonication is not carried out of alpha-synuclein fibrils, then in vivo they generally fail altogether, and in vitro studies they tend to have highly attenuated signals. The key parameters associated with best practices are taken directly from the reference cited below, which was put together by the Michael J. Fox Foundation, and are briefly concluded here. First of all, you have to sonicate your fibrils immediately before use, although the active component that is generated by that is unclear. It's thought to be essentially short fibrils. And the length thereof is cited as being 50 nanometers or smaller in order to generate phosphoserine 129 pathology. There doesn't seem to be a negative impact of extended sonication times or any differences in amyloid conformation or fibrillar structure associated with the process of sonication. However, short fibrils are generally deemed as being more efficient for seeding in vitro or in vivo experiments. One cautionary note is that sonication heat may negatively affect fibril stability, which may require some cooling. So in terms of the known effects of sonication, we know we reduce the fibrils in size to smaller segments, and the clumping that we get of fibrils is reduced, unsurprisingly. And there seems to be this somewhat mystical high activity or heightening of activity when the sonicated fibrils are added to cells or used in vivo. The question here for us was really, is something else happening here? So we went through a thought process. And one thing that is very important in that is that if we think back to that early slide of how the monomers aggregate together into oligomers and into protofibrils and finally into fibrils themselves, is that there's obviously a whole range of energetics and thermodynamics involved in these processes. And one key element that really sticks out is that the monomer forms are soluble. The oligomers are also in solution. In fact, the protofibrils or filaments are in solution as well. However, the fibrils are not. So there may be an unfavorable energetic component involved here of the substrate desolvation as these constructs are pushed out of solution. Now, the question for us was, if you could remove that limitation, essentially of a two-phase system, and thereby creating a single-phase system, if we were able to do that, we should see an increase in prion-like catalysis, such as the phosphoserine 129 activity or a THT response. And we know that that's what happens. So the question for us then was, well, does this sonication, which seems to have all the hallmarks associated with this process, create a single-phase system? Whereas typically we have two. We have a suspension and a solution. But what happens with sonication? Does it create a single phase, i.e. a fully soluble system? So we decided to test this. We took some fibrils in that start in suspension, and we looked at them under EM and measured their sizes, and we started to sonicate. And in this case, there's just a few micrographs here, and we start with 120 pulses at 20% amplitudes, and the size unsurprisingly decreases after that. That's then increased to 40% amplitudes, and again the size decreases. And this overall experiment is highlighted here on this bar graph, and as you can imagine, overall it's unsurprising that the more powerfully or the longer you sonicate, the shorter the fibrils become. That is known and is not new. However, it is a confirmation. So the question for us was, okay, what about the solubility of these? What happens if we look at the beginning and the end points of this reaction and look at the solubility? And there we begin to see some very interesting effects. So on the left-hand side of the control, you can see that the amount of protein in the supernatant is next to nothing, where almost everything is in the pellet. And that's exactly what we know to be true, because when we make the fibrils, they are in suspension, i.e. they are in a solid phase. However, at the maximum level of sonication on the far right-hand side, illustrated with the red arrows, you can see that after sonication, almost essentially all the material is now present in the supernatant and nothing left in the pellet, i.e. there is no pellet. So all the material has now been solubilized, and we have a single phased system, which will of course dramatically increase the ability of the molecules to interact with each other and their environment. The question then became well, does this last? Do they fall out of solution after a few minutes or hours or days? And so we looked at the solubility of these sonicated and unsonicated preparations after they had been stored at -80 degrees for 3 or 4 months. And again, in yellow, after for the sonicated materials, we can see that very little material falls back out of solution over the period of a number of months, whereas the same is also true, interestingly, for the supernatant. In other words, the material that was solubilized, even after the July to October time frame, stays in solution, having been frozen at -80 degrees. This is particularly interesting because it means that the original best practices note that these sonications have to be done immediately prior to the use in experimentation is not true. It means that these fibrils can be sonicated, the samples preserved and transported, and then used at a later time. So we decided to have a look at beta and gamma synuclein because, of course, the amino acid sequences are very similar with some core differences, and see how they responded to sonication. And interestingly, if we look at beta synuclein after sonication, we see a lot of it in the supernatant. But if we look at the control, we see that it was already in the supernatant. In other words, it was already overwhelmingly soluble. There seems to be a small increase in the level of solubility, but essentially it looks the same. The reverse seems to be true pretty much for gamma synuclein, where we get no change. So the controls on the left-hand side show almost no differences after they are sonicated. So what, therefore, is the mechanism? What is going on here? So we had to look at some of the sequences that differentiate alpha and beta and gamma synuclein. And you can see that beta synuclein is missing a large chunk of the domain that is highly hydrophobic in nature, that is associated with fibrillization. Now that's not new, and that is known, and that's also the reason why beta synuclein doesn't fibrilize on its own and needs to be treated really quite harshly in order to do that. But it may well be that just removing that hydrophobic stretch is simply enough for beta-synuclein fibrils to be sufficiently polar and interact with the solvent around them that allows them to be soluble without any extra help. Now, gamma-synuclein is a little bit different because it has that core area, but most of the differences are towards the end of the protein, the C-terminal end. And you can see here that I've highlighted some areas with red stars. Because my thought process here was, what happens when we sonicate? And sonication, although we think of it in terms of declumping, we think of it as, attenuating the size of these fibrils, is actually an extremely violent event that generates a lot of reactive oxygen species, including hydroxyl radicals. So these are areas that could be prone, or amino acids that could be prone to oxidation. And that hydroxylation of these moieties could be enough to increase the polarity of those stretches of the protein and hence make that fibril soluble in the case of alpha-synuclein. Now, bear in mind that the ones that I've highlighted are absent in gamma-synuclein, which is why we wouldn't see any effects when we sonicate gamma-synuclein. So in order to test this, we decided to do the following. A very simple experiment, we decided to take the protein fibrils and simply put them in an environment around pH 10. Now, you can actually make fibrils at this pH, that's not an issue, but we immersed it in that at 37 degrees for only 1 hour. And you can see on the left-hand side the control prior to this immersion at pH 10. But after just 1 hour, the fibrils are overwhelmingly in the supernatant, i.e. they have become soluble. So this is a primary indication that we're actually going through some hydroxylation processes here. So some covalent modifications that are creating a higher solubility of these fibrils. Moving on to fibril conjugations in the last part of this talk, here we have conjugated alpha-synuclein fibrils with a fluorophore atto 633. And once we had done that, we wanted to see if it exhibited a dose-dependent signal, and it clearly does not. So it's down the green line at the bottom. As we increase the amount of material, we get no signal, which would suggest that we hadn't actually carried out the conjugation. This led us to be quite suspicious, given the free standard curve in red. And so, given the highly structured nature of these fibrils, we decided to see if we could disrupt that, and if indeed we were getting self-quenching of these moieties. And indeed, this is the case. So if we disrupt the structure with SDS, all of a sudden we begin to get a dose-dependent signal, and we get fluorescence occurring and increasing with concentration. However, the same is not true for Triton, Tween, or other buffers. So the disruption needs to be quite severe in order for this to happen. Now, this is quite important because you can imagine that if you don't get your conjugation right, then you will not get a fluorescent signal despite the fact that the moieties are in fact conjugated. The other key elements, of course, aside from aspects such as sterility, are the fact that, of course, you want the fibril still to be biologically active. In other words, can it still seed reactions? And in this case, we were able to carry out these reactions and show that we were able to conjugate the fibrils at a level where the biological activity, despite being attenuated by 20% or so, were still active against the control. If we were to conjugate these particularly heavily, we can imagine, of course, alpha-synuclein being particularly prone to disruption by conjugation, that a high level of conjugation would eliminate the biological activity. This is also the case, for instance, with large tags such as GFP, when they are added to the monomeric structure during the production process, and then you get an absence of activity. So then we moved over to tau to see if we got the same story, and interestingly we did to some degree, but it was different. So here we do get a dose specific signal. As we increase those, we get more and more fluorescence versus the controls. However, if we do disrupt with SDS, we get an increased signal. So there is still a significant degree of self-quenching going on with conjugated tau fibrils in the same way as it is with alpha-synuclein, just to a lesser degree. However, if we look at the seeding capacity of conjugated tau fibrils, we get the reverse of what we see with alpha-synuclein. So the conjugated tau fibril, in this case, conjugated to tau ATO488, we actually see increased levels of seeding capabilities. And this may be associated with these somewhat hydrophobic fluorophores that are attached to the fibril, although that would just be supposition at this point. Clearly, the activity, however, can be maintained for the tau fibrils. Now, this would be particularly important in the use in general of conjugated fibrils, because it allows you to track where they are and where they go in terms of uptake, both in cells or even in vivo. And then if you can overlay on top of that the pathology generation in terms of, with tau, the phosphorylation of sites 202 or 205, or with alpha-synuclein with phosphoserine 129, you can begin to see how the seed is moving from one place to another and the effects thereof in a living system. The conclusions thereof of this webinar are that the monomeric, oligomeric, and filamentous or fibrillar protein constructs can have different properties from each other despite being made of exactly the same protein. The sonication of fibrils, in the case of alpha-synuclein preformed fibrils, results in fibril solubilization through a chemical modification, probably hydroxylation, and the fibril conjugation can impact typical fibril activities, but also lead to some complete self-quenching of signal. I'd like to acknowledge three people in particular: Alexandra Netter-Glangal, Sophie Sun, and Kenji Trotter, for doing the vast majority of this work in our laboratories. Should you have any questions, please feel free to send any inquiries to us. The information for our technical people and any information gathering that's required is on our website. You can email us directly at info@stressmarq.com with any questions. Thank you very much for your attention.